My name is Amanda Duchelis. I'm from the laboratory of Dr. Paul Vrana at the University of California, Irvine. Today I'm going to be showing you the procedure for oocyte retrieval. This entails isolating the oviducts from the female. Once the oviducts are isolated, we'll be injecting FHM media through the oviducts, and then oocytes should be exiting from the uterus. Once this is done, we'll isolate the oocytes in the media and use this for future experiments. What you might want to use this procedure for, in addition to isolating oocytes, is isolating embryos at specific time points. One reason you might want to see this procedure in the video format is because the different parts of the oviduct can be very hard to recognize. In this video, I will be pointing out the infundibulum where you will inject the FHM media. The dissecting tools that you'll be using are 2 sets of sharp forceps and 1 pair of sharp scissors. The oviducts that were taken from the female will be placed in FHM media, which is in a 35mm tissue culture dish. The eggs will be retrieved using a 30mm blunt end needle attached to a 1 milliliter syringe, and the oviduct will be stabilized with a sharp forceps. FHM is a holding media used to harvest the oocytes. KSOM is the media used to have the oocytes grow within the incubator if they become fertilized. Once the eggs and embryos have been retrieved from the oviduct, we will use a glass transfer pipette that has been pulled from a capillary tube, attached to some tubing, and mouth pipette those into some holding media. A glass concave dish is used to hold the embryos and eggs. They can be placed in each concave circle so that they are in a discrete location, which makes them easier to find. This is placed into this container so that it can be put into a CO2 incubator without using oil on top of the FHM media, or KSOM depending on the media. Water is put in the base, the top is closed, and the liquid stays in place. (70% ethanol is used to ensure the hair does not get in the way. We'll make a small incision in the abdominal region. An incision in the abdominal cavity. We'll enlarge the cut to expose the abdominal region. Once this is done, to expose the uterus, we'll move the intestines out of the way, and then the uterus is below. The uterus is Y-shaped in the rota, so I'll be taking part of the uterus. When that is cut, the oviduct looks like spaghetti. I'll be trying to take part of the oviduct. I'll take the oviduct and part of the ovaries, which is encased in fat. The other side is going to be taken at this point. Again, we're going to pick up the ovary, or the uterus. Pick up the uterus here, cut below our attachment point. The oviducts are found at this point. I'll be taking part of the ovary with us. This is then going to be placed in media. The uterus is still contracting. The uterus is made up of smooth muscle, so even though it's detached from the body, the neurons are still firing. The uterus is shown here.
while the oviduct looks like spaghetti, and the ovary is above that. The oviduct is not actually attached to the ovary, while we have only show a portion of the ovary at this point. My dominant hand is my right hand. In that I hold the 30 gauge blunt end needle. I use that as a forceps to isolate the infundibula. Once that is isolated, I will inject the blunt end needle into the infundibula and inject some liquid, which will flow out of the uterus. My left hand holds sharp forceps. This is used to isolate the infundibula and hold everything in place. The infundibula is a trumpet-shaped, isolated here. The tip of my needle is in the infundibula, which was pointing through the layer of tissue. Okay. To ensure that I've isolated all of the embryos and eggs, I like to squeeze like a tube of toothpaste on the uterus. Once you focus on the fat pads, you look for what looks like droplets of fat that are perfectly symmetrical. There's one right now just to the left of that long fat. That's a single cell egg. So what I'm going to do is take the mouth pipette, pick up the egg, put it into some more holding media so that we can isolate the eggs themselves.
Today I've shown you how to isolate oocytes from the female reproductive tract. In addition, I've shown you the different structures of the oviduct, in particular the infundibulum, which is the only structure in which you can inject the FHM media with a 30 gauge needle. With this technique, I've isolated oocytes and any embryos that have been present in the female reproductive tract. I can use these for gene expression studies or transgenic work.